Now, there was some interesting experiments that were performed several years back. They are called fusion experiments. Cells can be fused with some other cells. Usually a normal cell will be fused with a cancer cell. So, what do you expect if you fuse normal cell with a cancer cell? What do you expect usually? Cancer cells, right? But in some cases they have found a non-tumorigenic hybrid cells also. So, how this can happen? It is not only just enzymes, something in the normal cell tamed the tumor cell. That is why the hybrid cell which you expect to be proliferative. So, what could be the taming one? Could be a tumor suppressor. This is the example by which people started believing when the oncogenes were discovered. People were always telling that, like the Newton's third law I was telling you the other day also, there must be something opposite, you know. You can tell like that, but unless you do that experimentally and prove, people are not going to accept it. This is one such experiment which gave a clue that there are some factors in the normal cells which although have the property of taming the oncogenes. So, they named it as tumor suppressor genes. So, this led to the discovery of the tumor suppressor genes. Like if you remember, I told you the oncogenes have gain of function and they are dominant. So, as a result of mutation, whereas in the case of tumor suppressor genes, usually mutations result in loss of function of the genes. Mutation is generally in the form of deletion. Not only that, later I will also be talking about the tumor suppressor genes being inactivated by epigenetic alterations. Somehow the tumor suppressor genes are inactivated. See what happens in a cancer patient on the whole, he or she will have activated proto-oncogenes, that is the proto-oncogenes will be activated to oncogenes. This is not sufficient. They will also have inactivation of tumor suppressor genes. So, both will happen in the case of cancer patients. So, cancer patients will have activation of the oncogene as well as inactivation of the tumor suppressor genes. So, it is important to understand in each type of cancer what are the types of oncogenes that are activated, what are the types of tumor suppressor genes that are inactivated. There are many, obviously there are many oncogenes, many tumor suppressor genes. If we have an understanding of that, then we can design certain therapy to cure or treat cancer. So, that is the whole idea. That is why this whole molecular biology of cancer becomes important in that case. Are there any successful success stories? Yes. Recently also there was a very big success in the antibody therapy and there was a complete remission of the patients. Some cases later on I will again give that example that has come in the newspapers, that is considered to be a remarkable one. Of course, earlier also antibodies have been shown and people got Nobel Prize also for that. That is also there, but this one particular one was very interesting. We will talk about that a little bit later. So, all these things gave rise to the idea that there are tumor suppressor genes. The tumor suppressor genes basically do two things. One, they can inhibit cell growth or they can induce cell death, what we call as apoptosis. So, they can induce apoptosis or they can simply inhibit cell growth, both are different. One is simply cytostatic, the other one is cytotoxic. So, you have to understand that. You can completely eliminate or keep it, inhibit the cell growth for some time, dormant like thing, and then when the time comes, you can either activate or you can kill. When the situation is worsened, then induce apoptosis. So, it happens. Now, you may also wonder, many things have been discovered, for example, sometimes without doing the experiments or without doing the experimental work also. One such a thing is this Alfred Knudsen's two-hit hypothesis. he is not a cancer person actually, cancer biologist or anything. He was a statistics person. He was working at MD Anderson Cancer Center, Houston, where his job was probably at the medical record section, looking at the statistics of the various patients and all that. And he observed from his statistical he observed one important phenomenon, that you need at least 2 changes to get cancer, especially in children. He predicted that all of us inherit 2 alleles, ultimately you have one derived from the mother and one derived from the father, right? We have 2 alleles. And he said that in most of the sporadic, I told you what are sporadic cancers already, sporadic and hereditary. In sporadic cancers, he saw that there is a first hit and there must be second hit. 2 hits are necessary, ultimately you get cancer. If you have inherited some mutation in one of the alleles, you may be all right for some time, but if there is a second hit, then you get cancer. So, if you are having something, if you have inherited some gene which is going to predispose you into getting cancer, then you must have 2 hits. So, this is what is happening with tumor suppressor genes. Whereas oncogenes are dominant, even if one of the alleles get mutated, they will become dominant and you will have the gain of function. Whereas tumor suppressor genes are recessive, so you need 2 alleles to be hit. So, this is called 2 hit hypothesis. He just predicted that by looking at the statistics of various patients and certain genes. So, some such discovery was with RB, retinoblastoma gene, that was the tumor suppressor gene that was discovered later. There are number of tumor suppressor genes that have been discovered. One is called APC, adenomatous polyposis coli, in colon cancer. It is a tumor suppressor gene. The famous ones that you might have heard through newspapers are breast cancer related, what is called BRCA1 and BRCA2. BRCA1 and BRCA, breast cancer related cancer gene. Okay. So, that is BRCA1 and BRCA2, and then there are other number of them, but what is the most important tumor suppressor gene is P53. It is basically a transcription factor. Similarly, retinoblastoma gene. So, these are the different, tell you which one to remember. For example, you could remember this table. APC, I told you, familial adenomatous polyposis. In colon cancer, it is inactivated. Tumor suppressor gene means usually it is inactivated in cancer. So, BRCA1, BRCA2 obviously in breast and ovarian cancer. That is why, although I told you it is only 5% of the breast cancers you have hereditary, if you happen to have defects in these genes, then it is good for some checkup and do whatever is possible, because you are predisposed to getting that cancer. But that happens only in 5% of the cases, not in all. 95% is not, that is not happening. So, that has to be kept in mind. Similarly, there is one NF1 and NF2, neurofibromatosis type 1 and type 2. These happen in brain kind of cancers, neurofibromas, Schwann cells, meninges and so on. P53 by far is the most important thing. It is mutated in several cancers. The inherited syndrome happens in Lee-Fraumeni. Lee-Fraumeni syndrome it is called. So, these are the examples. Now, we will see some mechanisms. One example that I want to say is neurofibromin, NF1 as it is called. How it functions, how the tumor suppressor gene functions. It inactivates the oncogene, some of them, not all of them. Inactivates RAS soon after its stimulation by growth factors. So, allows for only transient activation of the RAS. So, this is what is happening. Mitotic signals will activate RAS. So, NF1 will, this symbol means it can block RAS activation. But when NF1 is inactive, this is another theory that you must remember. When you inhibit an inhibitor, you end up activating. So, in cancer, NF1 is inactivated. What is the function of the NF1? It inactivates RAS or it inhibits RAS. Now, in cancer, NF1 is inactivated. So, if you inhibit an inhibitor, then RAS is uninhibited, and so that leads to tumor. That is what is shown in this slide. I hope you understand that. Now, this is another important tumor suppressor gene, RB, retinoblastoma. Although the name is referring to retina of the eye and all that, it is present in all the cells, many cells. It is retinoblastoma. Of course, retinoblastoma refers to the condition of the cancer that is happening in the retina, but this gene is present in all the tissues, many tissues. It's a tumor suppressor protein. This is another example that I often tell people. Whenever something is phosphorylated, it is usually activated, but RB, when it is getting phosphorylated, it becomes inactivated. Usually, phosphorylation means activation in biology in many cases, but here phosphorylation is actually inactivation. How that happens, I will just tell you. Unphosphorylated RB negatively regulates the progression of the cell cycle into S phase. It does that by binding to what is called E2F. E2F is a transcription factor that promotes cell cycle, that actually triggers cell cycle. Now, RB goes and binds with this E2F and prevents it from working. So, cell cycle operation is suppressed. So, that is how RB acts as a tumor suppressor. So, that is a very important thing. So, in this complex, E2F is prevented from activating transcription of its target genes. E2F is a transcription factor. So, it will have its target genes which are involved in cell proliferation. So, that transcription is prevented once RB binds to it. During late G1, RB is actually phosphorylated by the cyclin D CDK4 complex, and it can no longer, once it is phosphorylated, it can no longer sequester E2F transcription factor. So, E2F is now free to go and bind its target, to go and transcribe its target genes which are involved in the S phase, synthetic phase of the cell cycle, and thereby transcription goes unhindered and allows the cell cycle to progress. So, this is the mechanism. I hope you understand. RB binds to E2F. E2F is a transcription factor that positively regulates the cell cycle. But when E2F is bound with RB, it cannot do its function. But when RB gets phosphorylated, it gets inactivated. Now, its interaction with E2F is interrupted, and now E2F is free to do cell cycle work. So, this is the major mechanism that you may have to understand. This is very important. Now, of course, more important one is P53. P53 is considered as a guardian of the genome. Why? Because whenever our genome gets some stress signals, or when our genome is disturbed by some signals, By the way, I have repeatedly told you that living cells do not do any function without a signal. So, when there are stress signals, p53 gets activated. So, it is a tumor suppressor that is activated in response to wide variety of stresses. It is not only in cancer, but in general also wide variety of cellular stresses. Those can be DNA damage, ribonucleotide depletion, redox modulation, oxidation reduction modulation, hypoxia, when you go kind of breathless, kind of when oxygen levels are going down, changes in cell addition, and several other stresses that can potentially activate oncogenes. So, in those cases it will try to, that is why I told you, when you have too much activation of oncogene, when you overexpress oncogene, it can result in cell death, I told you. Because what happens is, when such activated oncogenes are there, they will ultimately activate p53. Normally they won't, but when there is an excess, when there is an extraordinary condition, then p53 can get activated. I am talking about oncogenes, but otherwise p53 protein initially can induce growth arrest. It can also promote DNA repair, and it is one of the most well known inducers of apoptosis. p53 is very much connected to apoptosis. Later on, when I talk about apoptosis, I will talk about the mechanisms by which p53 ultimately induces apoptosis. By p21, all those mechanisms, we will talk about that. So, these kind of activities ultimately p53 is able to maintain the genomic stability. So, when the p53 is mutated, we have genomic instability, which is a characteristic feature of cancer. That is why p53 mutation, the more than, see, we talked about RAS mutation in 30%, whereas p53 mutation is more than 50% of the cancers. We will talk about it, it's coming in the next slide. So, elimination of p53 function leads to increased rates of mutation and resistance to apoptosis. p53 induces apoptosis. If there is p53 mutation, there will be resistance to apoptosis. So, that is how you have to understand it. That p53 is important in several biochemical pathways that are disturbed during cancer development. Consequently, mutations in p53 are the most frequent genetic changes that happen in human cancers. So, that is why p53 is extremely important. You can inactivate a transcription factor like p53 by several mechanisms. What are the mechanisms? Usually there is a point mutation, but there can be nonsense and splice mutations, deletion of one allele, overexpression of, again it is coming to the same, if you inhibit an inhibitor, it can become, MDM2 is its inhibitor, right. Actually what happens is, any MDM2 stimulates the degradation of p53, so that the levels are maintained at a normally at a lower level. p53 we do not want it a higher level in the normal system, only under some conditions you want it to be overexpressed, so that it can induce apoptosis. Because normally why do you want p53? If it is there, then it is going to kill cells. So, you do not want that, but under certain conditions you want the p53 to go up. So, normally it is kept suppressed by the so called MDM2. The human MDM2 refers to mouse gene. So, that is why it is written as HDM2, which is the human counterpart. It is the same HDM2. By gene amplification or deregulation, MDM2 if it is overexpressed, p53 will be underexpressed. So, that is the thing. So, you can inactivate p53 by several mechanisms. Then loss of p54, what is called ARF, that is another thing. Loss of function of upstream activators like ATM, CH1, loss of function of downstream effectors like bags. So many mechanisms are there. Post-translational modifications also can do that. Inactivation by certain viruses. For example, in cervical cancer, the HPV E6 can, there are other mechanisms also. E6 oncoprotein can inactivate p53. This is one of the most well known mechanisms in cervical cancer. There are what are known as E6 and E7 oncoproteins, oncogenes in cervical cancer. In that, E6 inactivates p53, whereas E7 inactivates RB. So, they do that. Viral infection can lead to inactivation of the tumor suppressor genes. So, that is one mechanism they can cause cancer. So, since it will be confusing to you, I have tried to give only 3 mechanisms, so that it is easier for you to understand. p53 is inactivated by at least 3 mechanisms. The most common event is mutation. That happens in more than about 50% of all human cancers, I told you. As with the other tumor suppressors, mutations can occur in somatic tissues or it can be inherited. Germline inherited p53 mutations give rise to what is called Li-Fraumeni syndrome, in which the affected individuals develop bone or soft tissue sarcomas. This is very rare, but that is one example. In addition, non-mutational, without mutation can you inactivate? There is a very interesting way by which p53 can be inactivated. After all, p53 is a transcription factor. So, what is a transcription factor? It will go on, usually proteins. p53 is also a protein, right? 53 refers to the 53 kilodalton. So, they have to go enter into the nucleus and bind DNA. This translocation, if you can prevent, that is, in other words, if you can retain them in the cytoplasm, they will not be active. If they do not go to the nucleus, how will they do transcription? That is simple. So, cytoplasmic retention is one mechanism by which it can be. This does not involve mutation. Simply retaining them, simply not allowing them to translocate to this one. That is one mechanism. Cytoplasmic sequestration, it is called. I have given it as last. But there is another, viruses can usually inactivate by forming certain inactive complexes. For example, just now I told you human papilloma virus E6 can inactivate. Other examples are the SV40 T antigen binds with T53 and forms inactive. This is happening with simian virus 40, SV40. Papilloma virus I already told you, by causing degradation of p53 through 26S proteasome. Under normal conditions, levels of p53 are kept by ubiquitination and degradation with the help of MDM2. That is what I told you. MDM2 performs this function by interacting with p53 at its N-terminal end, and then it targets the p53 for proteasome mediated degradation. So, that is the function of MDM2. This is how it, MDM2 and p53 function in a feedback loop. So, where activated p53 stimulates the expression of MDM2. MDM2 is a p53 is a transcription factor. One of the transcripts that are formed is its own inhibitor. This is another phenomenon that you must understand in biology. Certain transcription factors, they will transcribe their own inhibitor. Why? This is a feedback inhibition. They do not want excess of their own production. So, it is known as a feedback regulation. So, this is happening. p53 can encode for, can transcribe MDM2 because it is a transcription factor. One of the genes transcribed is none other than MDM2 itself. So, whenever p53 goes up, it will produce MDM2, which is going to degrade the p53, so that p53 levels can be brought down. So, that is the mechanism. This is important. Overexpression of MDM2 can suppress p53, and thereby it can, MDM2 can act as an oncogene actually, because it suppresses p53. In fact, people have found that MDM2 is overexpressed in osteosarcoma. I think we can kind of stop here. We have discovered, we have discussed the discovery of oncogenes, particularly SARC and ROS, I told you. And we listed names of few important oncogenes, molecular mechanisms by which proto-oncogene is activated to oncogene, and we listed tumor suppressor genes, and we have explained certain tumor suppressor genes, especially p53 and RB, how they function. I think we'll stop here.